Okay, hi, I'm David Kass, and I've never done this before, talking to a computer that's going to be presented, but I want to thank you for viewing my presentation. I would like to start with a funny joke, but I don't know any, so I'm going to get right into my presentation. So let's go here on my first slide. So you can see that the title of my talk is to generate authentic research experiences and forensic-based projects in undergraduate laboratory, which extends beyond just forensics, but being a forensic conference, I'm focusing on that aspect. Okay. Also note that we are a predominantly undergraduate institute, and the education part comes first, and that's why this is more an educational research project than a straightforward classic research experiment. Okay, so there's two projects, and both are designed to be consortium-based. So the projects focus on genomics, and genomics lends itself to being consortium-based, and the data can consistently be added as we develop databases to do these analyses. Okay, project one, I'll talk about the two projects. The project one is the development and use of an ALU-based tetraplexes for forensic ancestry and population genetics. And then project two will be the use of mammalian L1 sequences for generating a tree of life and also for forensic determination of unknown samples. Okay, so start project one, developing and using these ALU-based tetraplexes that I've been working on and designing. As background for educational settings, the most commonly markers that are used where students take a swab of their DNA and then do PCR and run an agarose gel is the D1S80 VNTR and the PV92 ALU dimorphous. And so those are the two main markers used. Okay, they're simple to use. D1S80 is the VNTR, got 16 base repeats. There's 29 alleles, so it's informative with 435 possible combinations. The primary drawback is that you have to run it on acrylamide gel and agarose gels, which students are more comfortable with. You can never get resolution to really determine the alleles. Okay, PV92 is an example of a SINE, which are short interspersed DNA elements. They're small, 75 to 500 base pairs. Okay. The ALU element is the primary SINE in primate genomes with over 1 million copies of them. Okay. These are retrotransposons, and I'll have a slide in a second, where these amplify throughout the genome, and that's where you got your 1 million copies, by a process of retrotransposition, which requires an RNA intermediate. These are RNA polymerase III transcribed, and they're non-autonomous. They require some other machinery. They don't code any proteins. And so here's a generic SINE. I'll show you. It has, I keep clicking the slide instead of the next thing. Okay, so here we go. So next, we've got the internal RNA polymerase III promoter boxes. We've got an A-tail, which is shown to be critical for the ability to retrotranspose. And most copies are not competent to retrotranspose. There's only a few so-called master genes. And you've got flanking direct repeats, which are found about 13 bases ranges, but they're at the ends. They're the product of the actual integration event because they're staggered nicks when they integrate into the genome. Okay, so here's kind of just a representation. You have a master gene, and you've got your enlarged view of it. So you can see TE. It's copied to RNA and reverse transcribed into cDNA. And so on the bottom, you have another chromosome that happened to be nicked. And then the copy, the cDNA is integrated into a new chromosomal site. So the second strand of DNA is generated, and now you've got a copy. So there's only a few master genes and over 1 million copies. Okay, so ALU element, here's an example of generating what's called an ALU dimorphism. The ALU dimorphism is where you simply have presence or absence of an ALU element. So it's a polymorphism, but there's only two forms, presence or absence. And as you can see on the slide, an ALU from the master gene, so there's the copy, can integrate into a certain chromosomal site. And then you have your derived form that contains an ALU and your ancestral form without the ALU. And so lots of individuals, including myself, have looked for the younger ALU elements, the ones that integrated more recently and are not fixed in the human genome out of the 1 million copies that are out there. So an estimated about 2,000 to 5,000 dimorphic ALUs in the human genome. And so what's nice about this, it's easy to use. And as you can see, if the ALU is present, you've got a forward and reverse primer, which are the same for both cases there. And you get 3 genotypes. Individuals could have both chromosomes have the ALU, could be heterozygous, or could be homozygous for not having the ALU, and it's easily identified on the 2% agarose gel. Okay, this is PV92, as I mentioned, the commonly used one, and you can see you get clear results, very simple to use, simple PCR reaction, crude preps of DNA from cheek swabs and run on an agarose gel. And we use gel red as a stain because it's not toxic like or cancerous like ethidium bromide is. Okay, but what I've done is take it a step further, and rather than use single individual ALU elements is to generate what I refer to as ALU tetraplexes, because you're looking at 4 ALUs at simultaneously in a single reaction. And so number 1, okay, that's an ALU at a certain locus, and 2 is a different locus. Okay, so we got these 4 different loci here. And the idea is to design primers that are spaced out where you'd be able to identify the variants on electrophoresis on agarose gel. Okay, so if we click here, you can see plus ALU and minus ALU for number 1. I show the arrows there. And then maybe for number 2, you can identify them there. And again, there's 3, and there's 4. And so the idea is that you can distinguish the 4 plus and minus ALU dimorphisms on the single gel. Okay, so how do I do this? First, you want to identify what's referred to as intermediate frequency ALUs and obtain that from the literature. The idea is you want both that are common. If one is rare, it's not going to be overly informative. Okay, then amplify individual loci, just get it to work. Identify heterozygotes, or you can take a homozygote and a heterozygote and mix them together. And then try doing 2 at a time. And for me, I tried all kinds of combinations, and what I found is using failsafe by Epicenter, which is now part of Illumina, works best with buffer ERI. They have a whole series of buffers. Okay, so then find pairs that work well together, fine tune them. Okay, if you've got a triplex already, then we go to step 7 to get the tetraplex. If you've got a duplex, then go back to step 4 to get the triplex. Okay, and then fine tune. Usually primer concentrations are a key thing. For one thing, band intensities can be different if it's heterozygous versus homozygous. And also the higher molecular weight bands usually are less intense than the low molecular weight bands. So things you have to consider when developing these tetraplexes. Okay, Oh, sorry, and it's a good idea to verify that these tetraplexes match up when you're just doing individual primer sets. Okay, so here's an example of the ALU tetraplex profiling system. Again, crude preps stored for several years. And so here is an example. Individual number 5 is heterozygous for locus 51. Okay, heterozygous for locus 182. Heterozygous for the ACE locus, and homozygous for lacking the ALU for the TPA locus. Okay, so I've presented the genotype for that individual. Individual number 6 is actually heterozygous for every single one and has 8 bands there. Okay, so if we want to try a paternity analysis using this technique, individual number 6 again is heterozygous, so uninformative, because you can't tell if got that band from the mother or the father. So here's a second tetraplex I developed, and again, number 6 is the mother. In this case, number 6 is homozygous for the 4 different loci. And the idea is to make sure that the children, all the bands in the children can be accounted for by the mother or the father. If they can't, then you can exclude the father. Okay, so 6 is the mother, 5 is the father, a potential father, and 7 and 8 are the children. The other represents another family. Okay, so 7 and 8, the 2 children have 90, or rather 9 plus, and so the father has 9 plus. Okay, so it's consistent. Okay, child number labeled number 7 got a band that's not present in the mother. Okay, which is 60 minus, but got that band from the father. And as we continue, number 225, which is an unusual situation, which I don't have time to talk about, but would gladly discuss. I have a paper on that one. And so child number 8 got that from the father. And over here, both children have the 50 minus allele, and dad contributed that. So father number 5 cannot be excluded. So it's a lot easier to exclude someone than, prove it. But in this case, it's all consistent. I also developed a third tetraplex. And by doing that, I've gone from 81 possible genotypes to 65,000. And when you include a third one, so 12 loci, we've got over 531,000 possible profiles. So again, thinking in terms of undergraduate classroom, it's a simple to use method, and you can generate very complex genotypes. Okay, and we compare to CODIS. You got the 13 short tandem repeats, and you can see the probability of a match is 10 to the minus 15th. What's interesting about ALU elements is that they're homoplasy-free markers. So if you have a G, it could, versus a C at a certain location, could be a reverse mutation, but these are homoplasy-free, so these have to be identical by descent because the odds of 2 integrating to the same spot are astronomical, and they don't excise precisely like they integrated. There are a couple of systems where you can analyze 30 markers at once and get close to the discriminatory power of CODIS. One would be to analyze each one of them individually, which is quite laborious and can be expensive. And there's a system out there that's pretty cool, based on flanking direct repeats, but requires the capillary system, fluorescent dyes, and so it's not the best tool for undergraduate classroom, although it's a really good tool. Okay, so the purpose of doing what I propose here, or what I've kind of started, is with the ALU tetraplex system, fewer reactions to do, fewer mini gels, so it's rapid, low cost, and if I'm working on a 4th tetraplex, and you can see you get 43 million possible profiles with that. Okay, so as a class forensic experiment, students would select a number from, I call it a hat, but a beaker or anything, a rolled up paper. So it's anonymous, and they only know what their number is unless they tell somebody else. Each student's given a swab and an extraction solution, and they label the tube with the number from their that they picked out of the hat. Okay, and they follow a protocol which is maybe like 15 to 30 minutes. It's not a very long protocol to do. It's crude prep. And you can put students into 3 groups, each developing a tetraplex master mix and aliquoting into numbered tubes, which they do. And then the student, let's say student number 1, will add there. So here's aliquoting shown in the figure. And student number 1 would put her DNA into tube number 1 for the 3 tetraplexes and individual number 2 and so on. And then as the instructor, I would choose one of these samples and place that as the unknown. And then we have negative control with no DNA. Okay, so here's the class forensic analysis. As you can see, you get quite a lot of variation in the profile. And then, okay, well, which is the unknown? And so can you identify the unknown? And it's clearly number 4. Number 4 is an exact match to the unknown, and it should be, because I picked number 4 as the unknown, which the students didn't know what number I picked. So this works as a nice class forensic analysis. We could do population analyses. There's a lot of data for these regarding population and allele frequencies. And so here's an example here. And we can look at genetic distances between populations. And here's from a paper where you've got Egyptian versus African-American populations for these various markers. And if you have 0.25 or higher, you have pretty high genetic variation between the populations. And there are 3 that are very high, particularly this ALU9 marker. Okay, and all of this has been part of the 1,000 Genomes Project, and Watkins' paper. There's a large number of populations they looked at for a lot of these ALU markers. So we already know their frequencies from a good number of populations. Okay, regarding Ancestry, because the Ancestry students just send their samples out and get some information, and there's no idea how that information was obtained, but this kind of shows a little bit greater understanding. But so if we assume the world population is in Hardy-Weinberg equilibrium, and a student had a particular genotype, one that I show you here, the chance of that genotype would be 1 out of 1,000,000. Okay, but when you compare different ethnic origins, if African-American ancestry, you would expect, based on allele frequencies, 1.17 times 10 to the minus 7 probability. Egyptian, in order of magnitude difference. Okay, so that profile is an order of magnitude more likely Egyptian than African-American ancestry. So it just gives you some idea. I mean, it's not totally foolproof accurate, but gives some idea. And along these lines, I've developed a website, and if anyone's interested, I've outlined here what the website is. And it models the Cold Spring Harbor website for the PV92, except this includes, at this point right now, it includes 8 of the ALUs, and you can compare the ALU elements, frequencies, Hardy-Weinberg equilibrium. You could make a class of population compared to another class, or I incorporated data from different populations, and you can compare class population to an actual population. Okay, so, but we're going to continue developing this some more as the project continues. Okay. Okay, so that was kind of it for project number 1. Project number 2, using L1 sequences, which is a member of the long interspersed DNA family of elements. L1 is found in mammals. They're found in all mammals. And the idea here is to bias amplification of young L1 elements and develop a tree of life and see if we can use it as a forensic determination of unknown samples. Okay, so the L1 has an internal promoter, but in this case, RNA polymerase II. And you have your 5' and 3' untranslated regions as you would for other protein encoding sequences. You have an open reading frame. It's actually called that ORF1, which is an RNA binding protein, and it's apparently cis-acting, so it will recognize its own RNA and allow for the retrotransposition process. And ORF2 is the protein that's involved with being an endonuclease and reverse transcriptase. Okay, there's also an A tail, and again, that seems to be important for the retrotransposition process. And again, the hallmarks of any transposable element is that when it integrates, it yields flanking direct repeats. Okay, so how do we determine young elements? Well, we kind of look at lots of L1 elements and develop a consensus sequence. And then we utilize what's referred to as concerted evolution. So in other words, when you're looking at lots of elements, they'll share certain sequences. So in this particular case, you have an ancestral L1 from an ancestral species, and it made copies. These copies accumulate random mutations. So that's what the X's are. They're all random mutations. Okay, and through time, the master gene, so that what's shown as ancestral L1 is the master gene that generates copies. Okay, you might have a variant within that master gene, and all the copies then, so species 2 has a diagnostic nucleotide N, and all the copies will have the N in them. Okay, and then the same for species 1 has, you can see a different N, I put it at a different position. And it'll still acquire random mutations, but the ones that are more similar to a consensus sequence, which represents the master gene, would be your younger elements. And so we've got species 1 would have an N at a certain position, diagnostic nucleotide, and species 2 have its diagnostic nucleotide. Okay, so we want to bias for younger elements, and the ORF2 region is the most conserved region. So develop primers based on the most conserved regions. And I'd have to use several slides to show you the whole L1, or even ORF2. So this is just a portion of the ORF2 sequence among different species, mammalian species. Okay, so develop different primer sets from that. And then the students, we had 12 DNA samples, and again, got them in groups and did the PCR. And so the first gel shows one primer set with 12 DNA samples and a negative control. And then the second one's a different primer set, and the third one is the third primer set. All of them were able to amplify products at the expected molecular weight. Okay, except the last one, there were a couple on the bottom there. We only see 2 and maybe a third one. So we chose primer set number 1 to continue with the study. Okay, and so continuing, what we did was shotgun clone them into a TA cloning vector and essentially generated these intra-L1 PCR libraries, because each band that you see is not a single locus. It represents thousands of different L1 elements found throughout the genome. Okay, so we generated these libraries, and we used blue-white colony selection, and then plasmid preps, and analyzed them on a gel to verify the size of the inserts, and then had them sequenced. And so this is one example of a sample where we had 5 different clones and developed a consensus sequence. And you can see there's very high similarity, that 99%, 96%, 97% to each other and to the consensus sequence. So it kind of verifies that we did bias for younger elements using these ORF2-based primers. Okay, now, what do we do with this regarding a tree of life in forensics? I'm going to start with the forensics. Okay, so the students, what they did was to get their sequences, and then each one of them, they called them, gave them a different clone name. So all clone number 1s, 1, 1A, 2, 1B, were from the same DNA sample, and then 2 and was from the same sample, and so on. So number 1, they got several clones, and when they did a GenBank screen, it showed the Norway rat, Rattus norvegicus, and then we tell them, okay, well, your sample was the rat. Number 2, it looks like it would be the rabbit, and indeed it was the rabbit. Number 3 looks like the dog. However, it was the fox. And then you might say, well, that didn't work out well, but there's a lack of fox sequences in GenBank of L1 elements, so it makes sense that it would find the closest relative. Same is true for the Aztec mouse, which is Peromyscus aztecus, not in the database, but we've got P. maniculatus and other Peromyscus species, so it corresponded to that. Sample number 10 was the dog, and it matched up to dog sequences. And then we had the chimpanzee, although in this case you've got human as well as chimpanzee, but chimpanzee did come out on top. And so forensically, they're able to use this and identify the source of the DNA sample that they got. And then secondly, developing a mammalian tree of life. And it's pretty consistent with what is known. And in fact, what's nice is all the rabbit sequences, you can see the blue circle there, clustered together. The house mouse sequences clustered, and with not lots of variation, a little more variation in the daegu and the fox. But as we keep developing primers, as we add more mammals, then it should, we expect it to come out more precise. In fact, both the carnivores and the primates are both, even though it might look like the carnivores are more related, when you look at the numbers on this, they're equally distant from the others. But the house mouse and the rat are related. They form a clade. The daegu and guinea pig are somewhat related, and they form a clade. Dog and fox, you can see the dog there is right in the middle of all these fox sequences. And the rabbit, it comes out just outside the rodents. So it's pretty much working out nicely. Again, there's lots of work to do, but so far so good. Okay, so what we want to do is generate a database of student intra-L1 sequences. And something we're working on, have a grant under review, and hope to continue with that. Okay, so to summarize, and I'm looking at my on-air time, and I'm doing pretty good, close to 30 minutes. The Alutexaplex system, we developed a simple, rapid, cost-conscious, accessible tool. And sorry, DNA is in lowercase here, but DNA profiling for the undergrad laboratory, and with several different applications that you can use it for. And what's nice is we can continue to develop a database, especially for the forensic part. Students just incorporate it, and eventually we find out allele frequencies of the entire population of individuals that have contributed in the profiles anonymously, of course. Okay. For the intra-L1 DNA PCR libraries, I've shown that we had biased it for the younger elements. It's a useful learning experience with a whole set of molecular tools, and there's a forensic component as well as overall generating a mammalian tree of life component. So anyway, thanks for listening to my presentation. Feel free to follow up with any questions via email. And again, thanks for listening.